Hello everyone. Today we have to discuss about nanobiotechnology for blood substitute. Okay. So here we have to discuss about how the nanobiotechnology can be utilized for the production of biomaterials that could be used for blood substitute. Okay. These are the learning objective of today's lecture. Okay. So first of all, after completion the discussion, students are able to understand about polyhemoglobin and present status of polyhemoglobin in clinical trials, and to know about the effect of tetrameric hemoglobin in polyhemoglobin on vasoconstriction and adverse cardiac effect. Okay. So, first of all, we need to discuss what is the nanobiotechnology. So, nanobiotechnology is the assembling of biological molecules into 1 to 100 nanometer dimension. Okay, so here we have to use biomolecules along with the utilization of the principle of nanotechnology to prepare biomaterials in the range of nano range, that is 10 to the power 9 10 to the power minus 9 meter scale. Okay? And these dimensions can be the diameter of nano-dimensional artificial cells, okay? Or any particle or membrane with nano-dimension thickness, okay? Or nanotubules with nano-dimension diameter. Okay, so anything which comes under the nano range could be studied under the nanobiotechnology. Okay, so nanotechnology utilization with the biotechnology along with the applications. Okay, in the field of diagnostics or therapeutics, we have to study in the nanobiotechnology. Okay, but this nanobiotechnology is a multidisciplinary approach which involve the various other types of techniques or sciences, okay, which utilize for the even some of the characterization techniques is a very important part of the study of nanobiomaterials. Okay, and the method of synthesis. Okay, like there are two types of methods there: top down approach, bottom up approach. Okay, so most of the time in the nanobiotechnology we have to apply bottom up approach. Okay, we have to prepare the nanomaterial from the atoms or molecules to arrange in the nano scale diameter. Okay, so when the nano scale diameter is achieved, okay, so these materials are called nanomaterials. Okay, so here we have to discuss the what nanomaterials could be used for the replacement of hemoglobin. Okay, and the first nanobiotechnology approach reported in the current scenario was the cross linking of hemoglobin into ultrathin polyhemoglobin membrane for artificial red blood cells. Okay, and if the emulsion is made even smaller, then whole artificial cell with their hemoglobin can be cross-linked into a polyhemoglobin of nano-dimension. Okay, so this is called polyhemoglobin, which is under the nano-dimension, and certain reagent to use for this conversion, like glutaraldehyde, can cross-link hemoglobin into the soluble polyhemoglobin of the nanodimensions. Okay, so this agent is used for the synthesis of polyhemoglobin, and a new generation of this approach include the nanobiotechnology assembly of hemoglobin. Okay, and some of the other molecule also used for the preparation of a derivative of hemoglobin that could be used in as a blood substitute. Okay, so like Catalase, Superoxide, dismutase into a soluble nanodimension complex. Okay, so the nanodimension is essential part of the preparation of material in the nanoscale, and this molecule, the polyhemoglobin, catalase, and superoxide dismutase, act as an oxygen carrier and an antioxidant for those conditions with the potential for ischemia-reperfusion injury. Wherever the required of blood transfusion is necessary, this substitute may used for the treatment. And another recent novel approach is the assembling of hemoglobin and fibrinogen into a soluble nano-dimension polyhemoglobin-fibrinogen complex. Okay, and this polyhemoglobin-fibrinogen complex also acts as a oxygen carrier with platelet-like activity that could be used as a blood substitute. And this is potentially useful in case of extensive blood loss requiring massive replacement using blood substitute. Okay, and resulting in the need for replacement of lead and clotting factor. And even nano-dimensional artificial cell can also be formed as a nano-dimension biodegradable polymer membrane. Okay, so these nano-dimension biodegradable polymer can be used as a artificial cell that have hemoglobin and RBC enzymes. Okay, so these are the various substitute which could be used in a hemoglobin as a hemoglobin or as a blood substitute. So first of all, we have to discuss polyhemoglobin. So polyhemoglobin. First of all, we need to know about hemoglobin. Okay, because polyhemoglobin prepared by the use of hemoglobin. So hemoglobin is a tetramer. Okay, it has a four group peptide in their structure. That is alpha one, beta one, alpha two, beta two. Okay, so this is called tetramer, and this tetramer break down into toxic dimer. Okay, and these toxic dimers are the alpha 1, beta 1, and alpha 2, beta 2. Okay, that cause vasoconstriction, renal toxicity, and other adverse effects if they are breakdown into their product. Even in the form of tetramer, hemoglobin molecule can cross the intercellular junction of blood vessel to cause adverse vasopressor effect. Okay, so this is the basic problem. We cannot use hemoglobin directly as a blood substitute. Okay, because hemoglobin found in the RBC, okay, an RBC membrane, and it acts as a oxygen and carbon dioxide carrier while it present in the embedded in the RBC membrane. And so some of the authors use the principle of nanobiotechnology to assemble hemoglobin molecule into a nano-dimensional polyhemoglobin. Okay, so to overcome these problem, these toxicity problem, okay, the researcher prepared the polyhemoglobin. So first, using the bifunctional agent, the subecyl chloride they use, and glutaraldehyde they use. Okay, so by the use of these agents, subecyl chloride and glutaraldehyde, the polyhemoglobin prepared. Okay, and the glutaraldehyde method was developed independently by the researcher for the development of hemoglobin-based oxygen carrier for clinical trials. Okay. The present status of polyhemoglobin in clinical trials we talked about. So it will undergo phase third clinical trials. Okay, and even phase third clinical trials have been completed on over 171 patients. That showing that this product can successfully replace extensive blood loss in a patient undergoing trauma surgery and other surgery by maintaining the total hemoglobin level 8 to 10 gram per dl. Okay, and this line needed for safe surgery with no reported side effect. Okay, so before undergoing any surgery, any treatment, it is essential to check the hemoglobin level. It must under the range of 8 to 10 gram per dl. Okay, so the transfusion of this polyhemoglobin in patient with hemoglobin level as low as 2 gram per dl can raise the hemoglobin level to with the 8 to 10 gram per dl when the patient is recovering from surgery. Okay, and normally the patient with hemoglobin level of less than 3 gram per dl do not survive. Okay, because they didn't have sufficient transport of oxygen and carbon dioxide. and some of the researchers have infused up to 10 liters of polyhemoglobin into individual trauma surgery patient. So, in United States, this product has been approved for the compassionate use in patient, and it is awaiting a regulatory decision for routine clinical use. And some other researchers have performed phase third clinical trials on its use in pre-hospital emergency, in which no typing and cross-matching matching is needed. So that is, it can be used right on the spot if there is loss, blood loss is there. So, given that the supply of hemoglobin from outdated donor blood is limited, so this is a major problem why we need the alternative of blood substitute. And glutaraldehyde cross-linking linked woman polyhemoglobin has been developed and tested in phase third clinical trials. Okay, so some of the researchers in the United States have performed a multi-center, multinational, and randomized single-blind RBC controlled trials in the patient undergoing elective orthopedic surgery, and a total of approximately 700 patients were randomized in a 1:1 ratio to receive either the polyhemoglobin or RBC at the time of first perioperative RBC transfusion decision. And of the patients receiving polyhemoglobin, 59.4% required no RBC transfusion on the way to follow-up, and 96.3% avoid transfusion with RBC on the first post-operative day, and up to 7.3% avoid RBC transfusion up to 7 days after surgery. Okay, so this is very good result has to be observed. And in the North America, this bovine polyhemoglobin has been approved for compassionate use in patient, and then even in South Africa, this product is approved for use in adult surgery patient to treat acute anemia and reduce allogenic blood use. Okay. So now we have to discuss what are the effects of tetrameric hemoglobin in polyhemoglobin on vasoconstriction and adverse cardiac effect. Okay. So in addition to the discussion on the polyhemoglobin, one from a human source and one from the moving source has to be taken under study, and other modified hemoglobins have also been prepared and studied, and these include intramolecularly cross-linked tetrameric hemoglobin and polyhemoglobin containing more than 30% of tetrameric hemoglobin that can cause vasoconstriction. And this had lead to the proposal that the intracellular junction of the endothelial lining of the vascular wall allow molecular dimension of hemoglobin to enter into the intestinal space, uh, and therefore hemoglobin acts as a sink in binding and removing nitric oxide needed for maintaining the normal tone of smooth muscle, and the result in the constriction of blood vessel and other smooth muscle, especially those of the esophagus and the gastrointestinal tract, has to be examined. And to test this, the researcher prepared polyhemoglobin containing different percentage of polymerized hemoglobin molecule using the same glutaraldehyde cross-link and characterized to ensure that they all have the same oxygen affinity. And the result shows that the polyhemoglobin with the lowest percentage of polymerized hemoglobin molecule does not cause vasoconstriction. Okay, so this is very good result because this is a major problem of hemoglobin, free hemoglobin breakdown. And with the increased percentage of unpolymerized hemoglobin molecule, there are increasing degree of vasoconstriction has to be observed. So this study clearly indicates that we can't use free hemoglobin, but we can use polyhemoglobin. And in red heart have high heart rate and they are more sensitive to ischemic changes. And the study by the researcher in the red show ischemic ECG changes in the form of ST elevation when they receive polyhemoglobin with a high percentage of tetrameric hemoglobin with even higher percentage of tetrameric hemoglobin, and there was increasing evidence of cardiac arrhythmia, and the ST elevation could be due to coronary vasoconstriction that results in decrease in the supply of oxygen to the heart, and this explains the occlusion of small subendocardial lesions in some primates and swine after the infusion with the one type of modified hemoglobin that consisting of 100% single hemoglobin molecule. So therefore, to avoid causing adverse vasopression pressure or cardiac effect, polyhemoglobin preparation must contain less than 2% of tetrameric hemoglobin. So this is very good observation, which could be helpful for the utilization of polyhemoglobin in as a blood substitute. Now, a new generation of nanobiotechnology blood substitute. So we need to talk about the new generation. So the assembling hemoglobin with catalase and superoxide dismutase has to be has potential to lose in substitution of blood. So as we know that the polyhemoglobin is likely to have an important role in certain clinical applications, even for the condition with the potential for ischemia-reperfusion injury. The use of new generation of blood substitute that is an oxygen carrier and an antioxidant should be considered. And the reason for this are that lack of oxygen supply in heart, sustained hemorrhagic shock, or stroke, or organ transplantation, and other conditions that may result in ischemia. That's why the ischemia lead to alteration in metabolic reaction and may produce hypoxanthine and activating the enzyme xanthine oxidase. And the level of hypoxanthine increase with the duration and severity of the ischemia. So when the tissue are reperfused with oxygen carrying fluid, the xanthine oxidase converts oxygen and hypoxanthine into superoxide. So by several mechanisms, superoxide result in the formation of oxygen radicals that can cause tissue injury. So that's why even in routine surgery, it will be important to rule out patient with a cardiac ischemia when using modified hemoglobin with no antioxidant. Otherwise, there could be adverse cardiac effect related to ischemia reperfusion. And even other conditions in which ischemia-reperfusion injury would be likely include and severe sustained hemorrhagic shock, stroke, myocardial infection, and organ transplantation. Okay, so for all these situations, the researcher used nanobiotechnology to assemble hemoglobin with catalase and superoxide dismutase into the soluble nano-dimension of polyhemoglobin, catalase, and SOD. Okay, so this is an oxygen carrier with the ability to remove oxygen radicals and superoxide dismutase and catalase convert to superoxide and superoxide into the hydroperoxide that is turned to converted into the water and oxygen. Okay, so it helps you to remove the half effect of free radical. So when we compare polyhemoglobin and polyhemoglobin catalase SOD, okay, it has to be the polyHb catalase SOD product remove significantly more oxygen radicals and peroxide and stabilize the cross-linked hemoglobin and resulting in a decrease in oxidation, iron, and heme release. Okay, so it would be more beneficial as compared to the polyhemoglobin. And close linking of these enzyme to polyhemoglobin is important because otherwise free SOD and catalase are removed rapidly from the circulation because half life of these SOD and catalase is less than 30 minutes. So in the form of polyhemoglobin catalase SOD, these enzymes circulate within half time more compared with the polyhemoglobin. so which is about 24 hours in human. So in the reperfusion of ischemic red intestine, the polyhemoglobin catalyzed SOD molecule significantly reduce the increase in oxygen radical that caused by the polyhemoglobin and a measure of increase in 3 to 4 dihydroxybenzoate. And some of the researchers also perform a study of global cerebral ischemia reperfusion in a hemorrhagic shock model. And this study was based on the bleeding anesthetized rat to hypotensive level, combined with the transient occlusion of both common carotid arteries after different length of time. Even this was followed by release of the occlusion of the carotid artery and reinfusion using different types of oxygen carrying fluid. So definitely the effect on the blood-brain barrier was followed by Evans blue extravagances and polyhemoglobin SOD catalase complex significantly attenuated the severity of blood-brain barrier disruption as compared to the use of saline stroma-free hemoglobin. Okay, and polyhemoglobin or a solution of free hemoglobin or SOD and catalase. Uh, in comparison with that, the complex of polyhemoglobin SOD catalase is shows higher effectiveness in this study, the brain edema was followed by based on changes in brain water content. So the changes in the brain water content of animals treated with polyhemoglobin SOD catalyst were not significantly different from those animals in the sham control group. Okay, so the increase in the water content of the animal treated with saline. So these hemoglobin and stroma free hemoglobin, polyhemoglobin, and the solution of free hemoglobin, sodium catalase were significantly higher than those of animal in the cell control, and the polyhemoglobin SOD catalase group by the fourth R and increase thereafter, and has significant under the significant level. With a p-value less than 0.01, and the attenuation in ischemia-reperfusion injury using polyhemoglobin SOD catalase shows significant promise for its potential role as a protective therapeutic agent in clinical situation of ischemia and oxidative stress, like stroke, myocardial infarction, sustained severe hemorrhagic shock, organ transplantation, and cardiopulmonary bypass. Okay, so these are the some of the area which the focus we need to focus on. And the next type of substitute that could be used as a substitute for blood. These are the polyhemoglobin-fibrinogen complex, which is a novel oxygen carrier with platelet-like properties. And in high blood volume loss, such as a trauma patient in hemorrhagic shock, who require massive blood transfusion, a large volume of RBC replacement alone does not replace platelets and coagulation factor. Okay, so this is the limitations of the condition, and resulting in coagulopathy and thrombocytopenia and promoting ongoing hemorrhage. So some of the researchers use nanobiotechnology to develop a blood substitute that is an oxygen carrier along with the other beneficial properties and with platelet-like properties. So this is a novel blood substitute, polyhemoglobin fibrinogen. Okay, and briefly, polyhemoglobin fibrinogen was prepared by the process of the fibrinogen solution. Approx 40 mg dissolved in the 4 ml of ringer lactate was added to the polymerizing polyHb solution. And 4 hours after polymerization began, after 24 hours of polymerization, the reaction was stopped by quenching with 2 molar lysine solution in a molar ratio of 200 is to 1 lysine to hemoglobin, and the solution was then dialysed against ringer lactate solution overnight to obtain the polyhemoglobin-fibrinogen complex. So in vitro experiment shows that the in which the glass tube were prepared with 250 microliter or 400 microliter of blood substitute and 250 microliter liquid of fresh blood were added to the 250 microliter liquid of blood substitute, and 100 microliter liquid of fresh blood were added to the 400 microliter liquid of blood substitute, and the timing was started with the fresh blood was added, and it has to be observed that with the polyhemoglobin the clot that formed did not adhere to the glass tube and no clotting time could be assessed, and on the other hand all of the clot that formed using polyhemoglobin fibrinogen stuck to the wall of the glass tube and could be quantified with a clotting time, okay, with the significant value. And in vivo experiment shows that the polyhemoglobin display clotting time similar to those polyhemoglobin fibrinogen for exchange transfusion of up to 80%, and beyond 80% exchange polyhemoglobin clot that form do not always stick to the site of the tube and would slide freely, and beyond 93% exchange no clot stuck for polyhemoglobin, and in contrast the clotting time for polyhemoglobin fibrinogen remain normal up to 98% exchange. So at which point there was only a slight increase in the clotting time has to be observed, and biodegradable polymer polymeric nano-dimension artificial red blood cells. So these complexes also can be used in as a blood substitute. So the researcher made use of his background in the use of the biodegradable polymer like polylactides for artificial cells containing hemoglobin and other biologically active material, and these biodegradable polymers are in routine use in surgery and drug delivery and other biomedical applications. And researchers are now using them to prepare nano-dimension biodegradable polymer membrane hemoglobin with a mean diameter approximately 80 to 200 nanometer. Okay, and these polylactides are degraded in the body into a lactic acid and then water and carbon dioxide. And for a 500 ml suspension, the total lactic acid produced is 83 milliequivalent, and this is far less than the normal resting body lactic acid production. And this is equivalent to the 1% of the capacity of body to break down lactic acid. Okay, so, in vitro experiment shows that bovine hemoglobin in these nano-dimension artificial RBC has the same P50 value, okay, and Bohr and Hill coefficient as compared to the normal RBC, and the content of hemoglobin can match that of RBC, and one can extract the whole content of RBC and then nano encapsulate this extract, and further additional enzyme can be added to the solution before the nano encapsulation process has to be began, and there thus additional superoxide dismutase catalase can also be included with the hemoglobin, and the researcher also used his background in artificial cell containing multi-enzyme protector recycling system to help solve the problem of methemoglobin formation. And in nano RBC, the biodegradable polymeric membrane can be made permeable to glucose and other molecules, and this allow us to prepare hemoglobin nanocapsule containing the methemoglobin reductase system in which external glucose can diffuse into the nanocapsule, and product of the reaction can diffuse out and therefore do not accumulate in the nanocapsule and inhibit the reaction. And in vitro study shows that this can convert methane globin to hemoglobin, And furthermore, reducing agent from the plasma can diffuse in to the nanocapsule to reduce hemoglobin to oxygen carrying hemoglobin. Okay, and in new experiment on rate shows that infused with one third of the total volume. And most recently, the researcher used a composite biodegradable polymeric membrane consisting of copolymer of polyethylene glycol with polylactic acid. And after extensive research using this approach, they have now prepared a nano-dimension artificial RBC that can retain their circulating hemoglobin level at double the duration of polyhemoglobin. Okay, so definitely whether you use nanobiotechnology, the existing polyhemoglobin effect has to be enhanced. And they investigated the long-term effect of polyethylene glycol and polylactic acid nano-artificial cells containing hemoglobin, and this is called nano RBC on renal and liver function also examined, okay, to check their harmful effect and spleen histological effect after one third blood volume top loading in rat. So the experiment that received one of the following infusion, that is nano RBC in ringer lactate, and SF hemoglobin, polyhemoglobin, or autologous red whole blood. Okay, so blood sample for biochemical analysis were taken before infusion and on the day 1721 after infusion. And rate were killed on day 21, and the kidney, liver, and spleen were excised for histological examination. And infusion of SF hemoglobin shows that it induces significantly decrease in renal function, as shown by elevated level of serum urea, creatine, and uric acid throughout the 21st day. And histological examination of kidney in the SF hemoglobin infusion group reveal focal tubular necrosis and intraluminary cellular debris in the proximal tubule. So in all other groups, there were no abnormality in renal biochemistry or histology has to be observed. So it would be conclude that injection of nano RBC did not have adverse effect on the renal function or renal histology. And nano RBC, polyhemoglobin, renal lactate, and red RBC did not have any significant adverse effect on alanine aminotransferase, aspartate aminotransferase, alkaline phosphate, creatine kinase, and amylase. So that's why the SF and HB induces significant adverse effect on the liver, and shown by the elevated alanine aminotransferase and aspartate aminotransferase throughout the 21st day. And on the 21st, the rat were killed, and the liver and spleen were excised for histological examination. And nano RBC, polyhemoglobin, single acted rat RBC did not cause any abnormality, has to be seen under the microscope, histological examination of the liver and spleen. So in SFHB group, the liver show accumulation of hemoglobin in central vein and sinusoids, and hepatic steatosis has to be observed. So so this experience shows that injected nano RBC can be efficiently metabolized and removed by the reticuloendocrine system, do not have biochemical or histological adverse effect on the liver or spleen. Okay. So what are the directions that using nanobiotechnology in the field of hemoglobin based substitutes? Okay. This discussion include some example to show the use of nanobiotechnology for the preparation of pellet substitutes. Okay, and the principle can be extended to other systems. And one example is the study conducted by the researcher of another soluble nanodimension complex of polyhemoglobin tyrosinase. And this substance has the combined function of increasing oxygen tension to sensitive melanoma to therapy and lowering systemic tyrosine to retard the growth of melanoma. And many other extension and modification of this general principle in nanobiotechnology are possible. So there is always the discussion for how safe are blood substitute, and it is reasonable to require that RBC substitutes and should be able to replace a allogenic RBC without causing more adverse effect than allogenic RBC. Okay. Uh, and one of the safety concerns regarding RBC substitutes is related to the vasoactivity. Here it is clear that not all hemoglobin-based blood substitutes have problem related to the vasoactivity, and such problems are only seen in those that contain a high proportion of tetrameric hemoglobin because it degrades to produce their two dimer, okay, which are toxic to and induce the cardiac problem and vasoconstriction problem. So the other potential problem is related to the inappropriate use of hemoglobin-based blood substitutes in those conditions that have potential for ischemia-reperfusion injury. So for this condition, one need to consider the use of polyhemoglobin, catalase, SOB complex as a blood substitute. Okay. If we talk about the precautions, certain precautions definitely followed, and some of the better hemoglobin-based blood substitute could be possible be safer that standard allogenic blood. And after all, recent research shows that liberal blood transfusion are associated with a 40% increase in mortality and 56% increase in the ischemic event when compared with a restrictive strategy. And the transfusion of short-term RBC is also associated with an increase in ischemic coronary events. So finally, it has to be shown that it is important for blood substitute to be as safe as allogenic blood, and it is not reasonable to require that RBC substitute should have no side effect, whereas standard donor RBC are associated with adverse effect, including ischemic coronary effect. Okay, so definitely, nanobiotechnology is the effective tool. Okay, so the first nano-activity approach reported in the discussion was the cross-link of hemoglobin into ultra-thin polyhemoglobin membrane for artificial red blood cell membrane. So even if the emulsion is made even smaller, then a whole artificial cell with their hemoglobin can be cross-linked into polyhemoglobin of the nano-dimension. So definitely glutaraldehyde can cross link hemoglobin into soluble polyhemoglobin of the nano-dimension. So a new generation of this approach include the nanobiotechnology assembly of hemoglobin with the catalase and superoxide dismutase into a soluble nano-dimensional complex. Okay, so this acts as an oxygen carrier and as an antioxidant for those conditions with potential for ischemia, reperfusion injury. Okay, so that's why. So now we have to summarize the lecture. So in this lecture, we have to discuss polyhemoglobin. Okay, which could be used as a blood substitution, and the second which we have to discuss here, the the polyhemoglobin with catalase and superoxide dismutase. Okay, this could be used. This is a nanobiotechnology based blood substitute. This could be used as a blood substitute. Okay, polyhemoglobin with catalase and superoxide dismutase. Okay, and in this lecture we have to discuss the third one, which is stroma-free hemoglobin. Okay, and the next one I have to discuss in this lecture, the polyhemoglobin-fibrinogen complex. Okay, which is also used as a substitute, blood substitute. Okay, and the next one we have to discuss in this lecture is biodegradable polymeric nano-dimensional artificial red blood cell. Okay, which use biodegradable polymer. Okay, like polylactides. Okay, so that's why these could be used as a blood substitutes. Okay, which can produce the artificial RBC by the use of biodegradable polymer, that is polylactides. Okay, and Okay, so that's why nanobiotechnology has a potential to produce other blood substitutes also. Okay, so these are the references for today's discussion. Now you have to attempt quiz. So first question is hemoglobin is monomer, dimer, or tetramer. Okay, so as we have to discuss in this lecture, hemoglobin is tetramer. Okay, so the correct option is A, tetramer. And hemoglobin molecule into nano-dimension polyhemoglobin by using. Okay, so how we can prepare polyhemoglobin? Okay, so in this lecture we have to discuss that silver chloride can be used and ultrathin can be used for the preparation of polyhemoglobin, which could be used as a artificial blood or blood substitute. And xanthosome is a high molecular weight hepatopolysaccharide that produced by the Xanthomonas campestris and polyanionic polysaccharide and has a good bioadhesive property. So all property related to the xanthosome, okay, which is a biodegradable polymer, and this could be used for preparation of artificial blood. And green synthesis of antibiotic can be done by the bacteria, fungi, plants. Okay, all of them used as for the green synthesis. And poly-Hb catalase SOD complex produced by the use of SOD catalase Hb. Okay, so definitely it could be used by the hemoglobin and catalase and a superoxide dismutase, all required for the production of poly-Hb catalase complex. Okay, SOD complex. And polyHb-fibrinogen produced by the using of use of the reagent. So it could be produced by the use of hydrogen and ringer lactate and glycine. Okay. So the correct option is B. So thank you everyone.